Hi, guys. I just want to pick up where I left off earlier today. And if you remember, we were talking about the definition of cancer, describing some of the traits that are consistent across a large number of cancers. Um, we gave some general characteristics, talking about cell division and the regulation of cell division and the inability to stop that cell division, and also that cancers are derived from abnormal cells. And then I made a few points. The first point was that all cells have the tools, at least, to become cancerous, even if we are talking about terminally differentiated cells. They do have the capacity to become immortal if they utilize the right genes. In addition to the fact that all cells have the ability to develop into cancer, or at least have the tools to develop into cancer, we talked about the categorization of cancers based upon their tissues of origin. I want to sort of pick up where we left off, as I said, by talking about the fact that, sorry, I have to share a screen here. Talking about the idea that cancers don't develop overnight, that all cancers develop gradually. And I said that cancers fall somewhere, or cells fall somewhere along a continuum, moving from what is a normal cell to one that is the gravest, shall we say, of cancers, and that is as an invasive cancer. I want to talk about how that process happens. So the first step along the continuum toward cancer, or toward invasive cancer, is a movement from a normal cell to a hyperplastic cell. And a hyperplastic cell is simply a cell that is overgrowing its normal boundaries. And I say that because there are two things that normally are required for a normal cell to divide. And these are, first, the adhesion to the extracellular matrix. And what we see in this hyperplastic lump, if you will, is that there are some cells that are not adhered to the extracellular matrix, but rather they are adhered to other cells. And the absence of adhesion to the extracellular matrix normally would stop a normal cell from undergoing a round of cell division. We call that anchorage dependence, or anchorage dependent growth. And that is a hallmark of a normal cell. And the absence of anchorage dependence is a hallmark of some cells that are moving along this cancer continuum. In addition to growing even when they're not adhered to the extracellular matrix, cells along this continuum are overriding something that we call contact inhibition. Cells normally would stop dividing if there were cells touching them on all of their sides. So contact with adjacent cells, that contact sends a signal to a cell that would cause it to say, hey, this area is full, and so therefore I should not divide and create any more cells. So the fact that these hyperplastic cells are overgrowing their boundaries says that they are overriding that contact inhibition, and they are no longer dependent upon their anchorage to the extracellular matrix. These are just a couple of images of hyperplasia. On the right-hand side, we see a gross image of gingival hyperplasia that I mentioned earlier in class, where the gums have overgrown. This does not mean that this is cancer, nor does it necessarily mean that it is a precancerous condition. It's simply overgrowth because of the absence of that contact inhibition and the failure of anchorage dependence. We see some microscopic images of some hyperplasia. Those of you who've taken histology can probably tell me what tissues these are from, but I don't know. But here we see, for example, cells that are growing into the lumen of whatever this lumen is. They're growing into the lumen, so they are hyperplastic cells. These cells look completely normal. The fact is that they are simply overriding those things that would normally stop the cells from undergoing rounds of division. So before I continue along the cancer continuum, I do want to diverge a little bit and talk about a condition known as metaplasia. Metaplasia is not on this simplistic drawing of the cancer continuum, but if you were to ask any thoracic or general surgeon, that person would tell you that metaplasia is also a precancerous condition. So metaplasia is the appearance of normal cells and the growth of normal cells in an abnormal location. Metaplasia often, and that's my dog, my puppy, playing with a squeaky toy, so I apologize. But that's what happens when you teach quote unquote classes from home. So metaplasias often are associated with areas where there are junctions between two different types of epithelial cell. So in this case, metaplasia occurs between the esophagus and the stomach, and we see that the appearance of secretory gastric cells within the esophagus. So again, cells that are normal, they're performing a normal function, but they're just doing it in the wrong place. And of course, we might imagine that secretion of stomach acid and digestive enzymes in the esophagus, where we don't have the same mucosal protection that we do in the stomach, that might be a bad thing. And maybe it's that injury rather than the metaplasia itself that's causing physicians to term this a precancerous condition. Another location where metaplasia is more frequently observed is the junction between the uterus and the cervix, for example. So I'll continue along my continuum, moving from hyperplasia to the next step along the continuum, and that is dysplasia. And as I mentioned in class, dysplasia is often categorized as a precancer. But as I also mentioned in class, we don't know what percentage of dysplastic cells are ultimately going to develop into cancer because physicians don't wait for that to happen. We know that there is some percentage of cells, dysplastic cells, that will develop into cancer. So rather than testing to see how many will, we just treat the dysplasia and or remove the dysplastic tumor so that it doesn't have an opportunity to develop into full-blown cancer. Here's a few images, and I'll move my head so that you can see them. A few images of examples of dysplasia. So right above my head is a pedunculated polyp that might be found in the colon. So this would be something that would be removed upon colonoscopy. Above that, we see an example of a pap smear, and we see normal looking cells, and here's the nucleus of a normal looking cell here. And here's the cell, and then the edge of the cell. And then we see some dysplastic cells sort of in the center of this image. I'll come back to those dysplastic cells in just a second because I want to look at the larger image where we see a junction between a normal area and a dysplastic area. In the normal area, we see epithelial cells, layers and layers of epithelial cells that are separated from the underlying stroma with the few cells that are in the stroma by the very clear basement membrane. And it's really clear in this image because, of course, it's outlined by a white dotted line. That same basement membrane is apparent in the dysplastic side, the right-hand side of this image, but we see a few things. So the basement membrane is still intact, but we see that the architecture of the epithelial portion of this tissue is a little messed up, right? There are many, many, many more layers of epithelial cells. And we also see what are odd looking or dysplastic epithelial cells that are highlighted by these white arrows here. I think that the hallmarks of dysplastic cells are probably more clear in the more magnified Pap smear image. And I'll mention just a few. When pathologists are looking for dysplasia, some of the things that they notice are a ruffled, shall we say, perimeter around the cell. So we see a uneven sort of cell membrane. If these cells were live rather than being fixed and stained as they are in this image, those ruffles or irregular edges would probably look like we call them blebs, but maybe bubbles, if you will, on the cell surface. So cancer cells tend to have these sort of lumps around the edges of their cells that when the tissues are fixed and stained, looks like these irregular edges here. And in addition to having irregular edges, dysplastic cells often have a higher number of nucleoli in the nucleus of them. So normally a normal number of nucleoli might be 3, 4, 5, something like that. So a nucleus would have 3 or 4 or 5 nucleoli, which are the sites of ribosomal subunit synthesis and rRNA synthesis in the nucleus. So you're going to have some lowish number of nucleoli within each nucleus, but cancer cells, dysplastic cells, have a higher number of nucleoli that may be associated with a higher rate of metabolic activity and protein synthesis. In addition to having more nucleoli, when we look across a population of dysplastic cells, we see that there's a greater degree of variability of the number of nucleoli in the nuclei of those dysplastic cells. So rather than having somewhere around 3 or 4 or 5 in all of the cells, we might have some cells that have 3, some cells that have 12, some cells that have 8, and some cells that have 19. So a greater degree of variability. Also pretty clear on this image of a Pap smear is we see that there is what appears to be a relatively large nucleus. And what I'm meaning to say is that the ratio between nucleoplasm and cytoplasm is greater. This larger nucleus per cytoplasm is a hallmark of dysplastic cells. This increased ratio of nucleus to cytoplasm may actually also play a role in a cell's metastatic potential, or at least success at being metastatic. And we'll talk about that maybe later in the semester when we dig deeper into the metastatic process. In addition to those hallmarks of individual cells, we see on the left-hand image that tissue architecture is often disrupted in dysplastic tissue. So it just does not look like normal tissue. And similar to hyperplastic tissue, in dysplastic tissue, we see a larger number or a larger percentage of mitotic cells. So there's a lot of mitosis going on. So that's consistent with the idea that cells become hyperplastic, they overgrow, so you have a lot of mitosis going on, but at some point that lot of mitosis produces some changes such that some cells become abnormal looking. And I just want to reiterate that dysplasia is often categorized as a precancerous condition. The next step along the continuum is cancer, in situ cancer. So in situ means in place, right? So this is non-disseminated cancer, but this is a malignant condition. The reason that this is considered cancer is because we see a breach, if you will, of the basement membrane that's underlying this particular carcinoma. As soon as a tumor begins to break through that basement membrane, that's when we know that we have a cancer situation. So this is a micrograph showing you the breach, if you will, of that basement membrane. The basement membrane is stained in a dark brown, and so on the right-hand side here of this image, we see a very clear dark brown line that represents the basement membrane. But on the left-hand side of this image, we see the absence of that dark brown line. And this suggests that the cells of this tumor, which are stained red, have somehow been able to degrade that basement membrane. And the degradation of that basement membrane then dissolves, if you will, that barrier between the epithelium and the underlying stroma, so that these cells can now, let's air quote it, escape from the primary tumor and enter the surrounding areas. So this is carcinoma in situ. I guess we've been looking at carcinomas, not only because carcinomas are the more common types of cancers, but also because when we follow this continuum, it's easier to see that continuum when looking at the presence or absence, if you will, of the basement membrane. When we're looking at sarcomas, obviously there's no basement membrane involved, so we have to determine cancer or where on the continuum we are in some other way. So the way that pathologists assess the degree of cancer or the where we are on the continuum of cancer, when we're talking about sarcomas, is pathologists will look for markers. Markers are proteins that are specific to a given type of cell. So as an example, we'll look at leiomyosarcomas, which are smooth muscle cancers. Normally, smooth muscle cells function as smooth muscle, and so they're going to be producing the protein smooth muscle actin. As those smooth muscle cells move along the continuum toward a leiomyosarcoma, their expression of smooth muscle actin is going to decrease and eventually disappear, such that those smooth muscle cells will no longer be identifiable, if you will, as a smooth muscle cell because they don't express smooth muscle actin. This, I'll call it a loss of self or a loss of identity that occurs in cancer is something that is termed transdifferentiation or sometimes dedifferentiation. So we're moving sort of backwards from being a specific and differentiated and terminal cell type to something that's no longer terminal and no longer determined to be a specific cell type. The most extreme example of that dedifferentiation or transdifferentiation is shown on this slide as anaplasia. And I would be willing to bet that even Dr. Myers, with his astute histological skills, would be unable to identify the tissue of origin of these anaplastic cells. Even if we looked for specific protein markers, they would be absent. So anaplastic cells are cells that have moved completely toward dedifferentiation and have lost all identifiable markers. It's extreme. A less extreme example of this trans or dedifferentiation is something that we term epithelial to mesenchymal transition, or EMT. This occurs when epithelial cancers or carcinomas lose their epithelialness, if that's a thing, and their epithelialness is usually very easily identified because an epithelial cell has an apical surface and a basolateral surface that are very different from one another in the proteins that are found there. When EMT, and that's, by the way, termed cell polarity. So we have an apical and a basolateral surface, and that's a polar cell. When a cell undergoes EMT, there is no longer cell polarity. In other words, we can't tell on these cells where the apical surface might have been and where the basolateral surface might have been. So these cells are no longer identifiable as epithelial. In addition to the loss of that polarity, another I guess, requirement for epithelialness is the expression of a cell-to-cell junction protein that's called E-cadherin, or epithelial cadherin. Cadherin proteins are responsible for cell-to-cell associations, holding the epithelial sheet together and holding that epithelial sheet together. And when cells undergo EMT, that E-cadherin expression is lost. And so the absence of polarity and the absence of E-cadherin are hallmarks of this epithelial to mesenchymal transition that, by the way, is a precursor toward the last step in our continuum, and that is to being an invasive or metastatic cancer. There are a number of steps that are required for metastasis to occur, and metastasis is the process by which some cells from a primary or original tumor are able to set up a, let's call it a, I don't know, auxiliary campus at another site. We have the primary tumor that is able to not only break through the epithelial barrier, but also to break through the basement membrane. As I said, that is potentially because it's acquired, cells have acquired perhaps expression of some protease or have somehow otherwise caused the degradation of that basement membrane so that cells can breach it. They move through the stroma and they break into either a blood vessel, as is shown in this image, or a lymphatic vessel to spread to a more distant site. In order for that to occur, the cancer cell has to cause, in this case, the endothelial cells that make up the lining of that blood vessel, cause those endothelial cells to separate from one another so that the cell can move through. And And in addition to that, the cells that enter the bloodstream have to be able to survive what I'm going to call are pretty hostile conditions within that blood vessel. If we think about the movement of blood cells through blood vessels, they're moving, you know, they're hauling, and they are banging into one another. And I'm just thinking of, you know, trying to merge onto a very crowded highway, not crowded so much that people are moving at like 20 miles an hour, but they're moving really quickly, and you have to merge into that traffic. And sometimes that's frightening. But these metastatic cancer cells have to sort of just pick up and move really quickly and be able to survive that beating that they're going to take in the vasculature, and not only survive that fast moving, bumping into one another situation, but they also have to somehow escape that situation. So they need to bind to the endothelial cells at a distant site, cause those endothelial cells to separate one another so that the cancer cell can leave, move through the underlying stroma, and then set up a secondary or auxiliary cancer at a tumor at a distant site. Okay, so there is a continuum along which cells progress in order to become cancerous. The next point that I want to make is that cancers are monoclonal, meaning that cancers or tumors are all derived, or all of the cells within a population of tumor cells, or of tumors, are all derived from a single cell. In other words, they're clones of one another, so that's a monoclonal tumor. So I have this in lighter font. I have, or are they, because I do think that there may be examples of polyclonal tumors. And that said, I'm going to give you several examples of monoclonal tumors that support the idea that all cancers are monoclonal, or that most cancers are monoclonal. The first, I'll call it piece of evidence, but it's not really evidence for monoclonality in tumors, is Occam's razor. So Occam's razor says, and I quote, "Entities should not be multiplied without necessity." That's a fancy ass way of saying that the simplest explanation is usually the right one. For those of you going into clinical careers, you will hear this very often. Often it will be associated with something like, you know, if you hear a quack, it's a duck, or if you hear the sound of hooves, it's probably a horse and not a zebra, those sorts of things. That's Occam's razor. And so the simplest explanation for tumor development is that we have only one cell that in which things go awry, versus having a number of cells in which different things have gone awry, but those rogue cells have all sort of clustered together to create a gang of cells that is a polyclonal tumor. So Occam's razor is the most simple explanation, and that tumors are monoclonal. But here's the deal. When we look at tumors, they are not homogeneous. They are, in fact, very heterogeneous. So there are different subpopulations, if you will, of cells within any given tumor. So if tumors develop monoclonally, how do they become heterogeneous? I think you know the answer already, but we'll come back to that in just a second. There is a lot of supporting evidence beyond Occam's razor for the monoclonality of tumors. I'll give you three. The first of them requires a a review of what bar bodies are. So if you remember, females have 2 X chromosomes, one, I'll say it's the blue one in this image, that has been inherited from dad, and the other, the pink one in this image, that has been inherited from mom. 2 X chromosomes is one too many. We need to turn off transcription from one of those X chromosomes, lest we have too much product from the 1800, for some at least, of the 1800 genes that the X chromosome carries. So we inactivate one of our X chromosomes by packaging it very tightly with proteins to create what is called a bar body. This inactivation of an X chromosome is random, meaning that in some cells, we're going to turn off the mom chromosome. So that's in the lower part of this slide. And in other instances, we're going to turn off the dad X chromosome, and that's the top part of this slide. And then when those cells undergo mitosis, it produces a tissue that is a mosaic of cells, some of which are transcribing from the maternal X chromosome and others of which are transcribing from the paternal X chromosome. So, as I said, the X chromosome carries about 1,800 genes, and among those 1,800 genes is a gene for glucose 6-phosphate dehydrogenase, or G6PD. This is an enzyme that is involved in the production of energy. So there are 2 different versions, if you will, of this G6PD gene. Both of those versions function normally, although they behave somewhat differently, but they function normally. So an individual we cannot call one of them mutant because they both function normally. So we call these different alleles polymorphisms or isoforms or versions of the gene. Although these gene products, these different gene products, both function normally, as I said, they behave a little bit differently, such that one of them is more heat resistant than the other. So here I have an image of some intestinal epithelium from an individual who is heterozygous, containing both alleles, some of which are heat resistant and some of which are heat labile. So the epithelial cells that stain a darker blue are those that are heat resistant. So this is an epithelial sheet that has been exposed to heat. So this G6PD is still active, and the heat labile G6PD is where the lighter blue regions are. And so if it weren't heated, it would be functioning normally, but now we have a difference in functionality because of that heating. And we can also sort of look at the mosaic nature, if you will, of this epithelial sheet, where we see stripes or spots where maybe all of the cells in this dark blue stripe are descendants of a single cell that had the heat resistant form of the protein, and all of the cells in this sort of light blue stripe are derived from a single cell that had the heat labile version or isoform of the G6PD protein. Okay, so we have these 2 different isoforms, and they both function fine, although we are able to distinguish them. So we know that 30% of African American women are heterozygous for the 2 different versions, if you will, of this G6PD gene. So they have those, they are like what this epithelial sheet shows. In addition to being having a difference in heat resistance, these 2 different isoforms migrate differently on an SDS page. So for those of you who've had biochemistry or other courses where you've done SDS page, you know what this is showing. But for those of you who haven't, SDS page separates proteins based upon their molecular size. When we look at the left-hand sort of lane here, we see what it would look like if an individual had one version, let's call this the heat stable form of the G6PD gene. And this person is a homozygous because they only have that form. Similarly, the center lane shows what a homozygous individual's profile might look like if they only had the heat labile form of the G6PD gene. So they would produce only one type of protein that would migrate at apparently a higher molecular weight. But a heterozygote individual is going to have both versions of the gene and therefore both versions of the protein. And those are going to separate based upon their different molecular sizes using SDS page. Finally, when we look at tumors from heterozygous individuals, we see that the tumors are all homozygous, even though the patients themselves are heterozygous, meaning that the tumors arose from cells that had the same X chromosome inactivated. Now, it's possible that two different cells by randomness had inactivated the same X chromosome and had come together to form that gang of cells that is a tumor. But it's more likely that all of the cells within a given tumor had arisen from a single cell that had some random X chromosome inactivated. In this case, it doesn't matter whether it's the heat stable or the heat labile version of the G6PD gene that's expressed. That's not contributing to the development of cancer because there are tumors that have either, but not both. A second piece of evidence to support the monoclonal nature of tumors comes in a specific type of blood cancer called multiple myeloma. Multiple myeloma is a B cell cancer or cancer of B cells. And this slide mostly is showing you how we make B cells. So for those of you who've had immunology, you know that B cells are responsible for making antibodies. But before those B cells make antibodies, they express something that's called a B cell receptor on their surface. And this B cell receptor is effectively a surface-bound antibody. Each B cell is going to have a different version, if you will, of B cell receptor. And that's sort of depicted in the center image where there are a bunch of B cells and they are displaying, shall we say, different B cell receptors, and that's depicted by different colors of B cell receptors on their surfaces. When exposed to some sort of antigen, the B cells that are able to bind to that antigen are able to, they become activated, and that activation does a couple of things. The first thing that that activation does, and I'm going to move my head here, the first thing that that activation does is it causes reproduction in those B cells. So now you're going to have a lot of B cells that are displaying a specific type of B cell receptor. So when we look at the blood of an individual who's had this amplification, we see a lot of these B cells that are similar to one another. And in addition to reproducing and creating a lot of B cells, we are also going to begin secreting those antibodies. So rather than being membrane bound, now some of those membrane bound B cell receptors become soluble antibodies that are going to be found in the blood. And that's through the maturation of the immature B cells to plasma cells. So, okay, so if we look at the blood of patients who have multiple myeloma, not only do they have a lot of antibody because they've got a lot of B cells in their blood, but they also have a lot of the same type of antibody, a monoclonal antibody, suggesting that it was a single B cell that over reproduced to produce the multiple myeloma cancer. The third and final example of monoclonality in tumors that I'll share with you is another hematopoietic cancer, and this is chronic myelogenous leukemia, or CML. So shown at the top is a depiction of normal tissue where only one cell within that normal tissue is a little bit weird. And the way that that one cell is a little bit weird is it has experienced a chromosomal translocation. That little yellow leg or arm, if you will, of the chromosome is meant to be the arm or leg of a different chromosome that's not shown here. So a portion of the yellow chromosome was transferred to this chromosome right here. And at the same time, the red leg that used to be there got transferred to the yellow chromosome. So that's a chromosomal translocation. When we look at the cells that comprise a CML cancer, all of those cells have chromosomal translocations. Chromosomal translocations are in and of themselves very rare. But in addition to having chromosomal translocations, all of the cells within this CML have the same chromosomal translocation. And that seems very highly unlikely that multiple cells would experience chromosomal translocations in exactly the same location. So once again, the presence of this type of cancer in which all of the cells have the same chromosomal translocation, that's very strong evidence for the monoclonal nature of cancers. So I've just given you 3 examples of monoclonal tumors. So what we're observing is, or what I've spoken about is what's shown on the left-hand side of this slide. But does that mean that all cancers or all tumors are monoclonal in nature? I don't know that we know the answer to that, but it is the simplest explanation. In order for us to use that simple explanation, however, we have to consider how the heterogeneous tumors that we often observe in individuals have arisen. And as I said earlier, I think you know the answer to that, but I think it's how every population sort of diverges into subpopulation, and that is essentially evolution. I mean, within a tumor, there are going to be different microenvironments, and cells are going to learn to adapt to those different microenvironments. And because of that, we are not going to see all of the cells within, especially a pretty large tumor, be all identical to one another. But I will throw out some what ifs. Is it possible that the monoclonal tumors that I just spoke to you about, is it possible that that they arose from a polyclonal tumor, this gang of rogue different cells, so that only one type remained? by the time we looked at it. In other words, in that could a polyclonal tumor have developed, and the purple cells within that tumor and the teal cells and the brown cells within that tumor were all outcompeted by the green cells, so that now we have a monoclonal tumor that we're detecting in these examples that arose in a very different way. I think that's certainly a possibility. Another possibility is that the tumors that we're looking at a single thing, like G6PD production or chromosomal translocations. And maybe if we looked at other things, they wouldn't be monoclonal. Maybe our tests to distinguish different cells within what we're calling a monoclonal tumor are not sensitive enough for us to see those differences. So I guess I will leave it at that and say that I've just given you 2 new points to round out or layer upon the 2 that we spoke about in class. And when we come back to class on Wednesday, virtually via Zoom, we will talk about 2 more and 2 more distinguishing features of the majority of cancers. And then hopefully we will be able to move on to talking about the different types of cancer genes that contribute to the development of cancer. Okay, have a good weekend, and we will see you next week.